Hey everyone, welcome to Valcrion by Team Queens Canada, a transdermal biosensor to detect critical metabolites. We started with a patient, not an idea. Our team is from Canada, and in Canada there is a very prevalent disease that affects about 12.5% of the adult population. Recent estimates are at about 4 million Canadians affected, and there is a rising incidence of end-stage kidney disease. This makes this disease very severe and very prevalent in Canada. This disease is chronic kidney disease, and 1 in 8 adult Canadians are affected by it. So what is chronic kidney disease? It is defined by a progressive loss in kidney function over time. But it doesn't just affect Canadians. In fact, it is very prevalent worldwide. About 1 in 11 people globally suffer from chronic kidney disease, putting this at 697.5 million cases worldwide. CKD is so severe that it causes more deaths than breast cancer or prostate cancer. In fact, over 1 million people die annually due to CKD, which is a 33% increase over the past decade. This makes CKD the 12th leading cause of death worldwide and 10th in Canada. So expectedly, CKD is associated with a tremendous economic burden. In Canada, where the population is about 37 million, we spend over $40 billion annually on CKD. So what makes up the $40 billion of annual spending that we do in Canada? Well, there are two main pillars of the economic burden. One, we have treatments, and two, we have diagnostics, including things like metabolite assays and monitoring tools. Given how much we spend on diagnostics, it gives the impression that there is little left to be desired. However, there is one major problem. And that is that currently there is no tool, method, or system in place at the moment to frequently monitor CKD metabolites. This is a problem because as your glomerular filtration rate, or GFR, an indicator of kidney status, goes down, your frequency of cardiovascular events goes up. This means that chronic kidney disease patients have a high risk of developing cardiovascular disease. And abnormal mineral metabolism is a very important risk factor for cardiovascular disease. Abnormal mineral metabolism in CKD can be thought of in 3 stages. You have biomarker abnormalities, bone abnormalities, and vascular calcification. Some of the most important things to monitor are biomarker abnormalities, because you can see elevations in parathyroid hormone, elevations in phosphate, elevations in potassium, and elevations in fibroblast growth factor, or FGF23. High levels of phosphate can lead to vascular calcification, which can take a healthy blood vessel like this and turn it into one that looks like this, and this will make individuals more prone to cardiovascular events. Alterations in levels of PTH can lead to progressive bone loss, taking one that is healthy to one that is thinned and weakened. Elevations in levels of potassium can lead to an abnormal heart rhythm, making an individual more susceptible to cardiac arrest and death. And to make matters worse, in modern fast food industry, sodium in food products is being replaced by phosphate in an attempt to make them healthier. The recommended daily intake of an adult is 700 milligrams of phosphate, yet each of these items have very high phosphate counts that aren't disclosed to the public. So this highlights how important it really is to keep track of your phosphate, potassium, FGF, or PTH. Yet, how often are we monitoring CKD in Canada? Well, blood is only sampled once every 6 weeks, making this a major problem because things like phosphate, potassium, FGF23, or PTH, they fluctuate on a daily basis. Therefore, there has to be a system in place to measure these analytes frequently in order to provide patients with the best possible health outcomes. So clearly at the moment there's a large gap between patients and clinical practice to provide patients with a tool for frequent monitoring of their CKD status, and our project, Velcrion, aims to bridge this gap. We will do this by creating a biosensor that measures in vivo levels of phosphate, potassium PTH, FGF23, and glucose in order to provide patients with the best possible health outcomes.
We set out with a number of goals for a system that could track the biomarkers of patients with chronic kidney disease. Our system would have to deliver reliable measurements for clinical purposes. We needed a device that could detect biomarkers in the micromolar range found in the human body, something that could be reusable, constantly redetecting biomarker concentrations, had to be fairly priced. Now, while we aren't in the stage of selling our device, we sought to use affordable parts, especially for people that are uninsured. And finally, our device had to be safe, meaning nothing would enter the body or cause bodily harm. Safety is really important to us. Even though we cannot make it into the lab because of the coronavirus this summer, our protocols have safety in mind. We use lab-strain E. coli cells to express and purify our proteins. Now, normally all of our wet lab team would undergo significant training. However, this year our team was focused on ensuring that our workspace was safe and ergonomic. Working from home can be just as tiring, and so we aim to get around this by promoting fun group meetings and regular breaks. Our device uses a variety of proteins, and we're going to share that with you. It became apparent, however, that we would need to immobilize these proteins in order to have them reusable. Current methods do exist. For example, on the left you'll see a snap tag, and this is from New England Biolabs, and that could bind to a target and a device surface. Or we could use something like an alkyl halide chain, and that'll bind a gold plate and a cysteine. Now, these methods are great, but they're very expensive, and due to the costs of synthetic synthesis, might be too high for the consumer. More importantly, these methods, especially the alkyl halide, could irritate the skin, and we knew that for a device that was so close to the skin, we would need a different approach. Now, in order to monitor in vivo levels of ions like phosphate and hormones like FGF23, at the core of our biosensor is a fluorescent binding protein. And we have glucose binding protein depicted in wheat color, and bacterial protein that binds glucose with high specificity, which undergoes a conformational change. When ion like glucose interacts with this binding protein, the neon green and cherry fluorophores come together and an increase in intensity is observed. Of course, we could directly correlate this with the concentration of glucose. Our construct here is anchored through a coiled coil system, and that adheres to the gold plate of our fluidic chamber. Here we have one coil cysteine tail, and that's interacted with the mutated cysteine on the glucose binding protein used to anchor the construct. Our opposing coil cysteine tail end binds the gold surface of our biosensor, and these are strong hydrophobic interactions, and electrostatic peripheral charges are associated the two coils together. Now, here in this video, our phosphate binds bacterial phosphate binding protein, and the protein actually goes a hinge-like conformational change. And the conformational shift between the apo and holo protein is great enough here that it can move the fluorophores together. Now, this proof of concept is an alignment movie, where we can view the change between bound and unbound phosphate binding protein. Our fluorophore choice, of course, is very purposeful. Conventional fluorophores like GFP or RFP, while they are similar in structure to our choices, they've been refined to fluorophores with greater intensity and range. As a team, it was decided that to have the most advanced device meant having the most advanced fluorophore. And here we have our donor fluorophore, M neon green, and this is a beta barrel structure. Our acceptor fluorophore, again a beta barrel structure, this time with a higher emission wavelength, is M cherry. Both of these are similar in structure and they've been well tested together as a pair. Now our neon green and cherry pair has a good overlap in their spectras, and this gives us some great intensity. It's very bright, and importantly, we have an extended dynamic linear range when compared to conventional GFP or RFP pair. Its emission is also visible at about 612 nanometers, which puts it in the visible light spectrum. Now, to ensure that our coiled coil cysteine tail binds to our protein in the right orientation, we have to remove every cysteine except for one, and that's opposite to the active site on the surface of our protein. We've done a lot of research with our constructs to ensure that this attachment doesn't hinder fluorophores movement or ligand binding. And we've run intensive homology searches to ensure that our substitutions wouldn't affect our protein structure. And what's great about this technique is that using an E. coli chassis, we can always ensure that the binding protein uses the same high intensity fluorophore tail pairs. We've done this successfully for almost all of our constructs. Now, one method for this homology searching is to use the FIRE2BLAST method. FIRE2, if you don't know, it's an online protein homology server. This will compare your structure to all the structures in the protein databank, and it gives you an output with alternative residues at each amino acid position. For example, if I wanted to remove a cysteine, I can see which residues are commonly found at that position. Based on the protein's 3D structure and this information, I can make a highly informed conservative mutation. And that's exactly what we did with our proteins. We removed free cysteines in the protein and mutated in a cysteine residue for anchoring. Noticeably, our free cysteine residue is on an ordered secondary structural region, away from the active site, and it faces the surface of our protein. This is for all of our constructs. Now that fluorescent binding protein has to be anchored. Our coiled coil tail binds to our cysteine residue on the protein through a strong disulfide bridge. Our 5-heptide repeated alpha helix coiled coil has a tough hydrophobic core and electrostatics. It's so strong that it's actually heat and protease resistant. And we were able to perform modeling of the interaction of the coiled coil and the binding protein. This was to ensure that in 3D space there was no steric clash. The other cysteine tail end of our coiled coil can form a strong interaction with the gold chamber of our biosensor, ensuring accurate detection. And you can even see how tight that interaction is. And with our electrostatic potential map, it really highlights just the difference in charges of the coils and a potential for interaction based on that alone. We also performed other modeling using Chimera and Pymol. These are two visualization tools. The focus of this, of course, is to ensure that there's no steric hindrance between the fluorophores and the binding proteins, as this might affect the function or assembly. We used both cartoon models for display purposes. A ball and stick model, pictured on the right, you can see all the residues in 3D space. Electrostatic modeling was also performed. With this, we can ensure we aren't blocking the active sites of our proteins with our fluorophore pairs that we added. Now, some of our proteins, like the ones that bind hormones, for example, sometimes they don't undergo a large enough conformational change. For example, parathyroid hormone receptor. In this case, we have an abundance of PTH tagged with one fluorophore, bound to the PTH receptor tagged with the accompanying fluorophore. And the intensity would be constant. Now, as PTH from the body enters the system and competes for the receptor binding, the fluorescently tagged PTH is removed from some of these binding sites. A system like this would be similar to a competition assay. So as the PTH competes with the tagged PTH, a drop in intensity is observed. Now, this new intensity would correspond to a concentration of the particular biomarker, in this case PTH. To express our protein of interest, we use the PET24D vector. We insert that into our E. coli cells, to grow the protein with the T7 polymerase. This has a C-terminal his tag, which makes purification much easier. And the DNA for the plasmids, with their FRET fluorophores attached, are transformed. We use the restriction digest XHOI and NCOI. Now, smaller proteins like our coils are a little more difficult to express. We insert them into our plasmid with the GFP attached. This makes it a lot easier for the cells to express the protein properly. In between the GFP and the coil, we have a TEV protease linker. This is the amino acids ENLYFGQ. Now, when the TEV protease cuts at this site, it gives us a nice free coil to work with. Purification is very simple, of course, because we use the histag method. Here we use a nickel chromatography column, which the cell lysate is put into, and this has nickel lining it. Imidazole is flowed through the column to compete with the histidine-tagged proteins and their affinity for nickel. Latter fractions are collected, and these would likely have the purified protein. We can confirm this with SDS page, and if we have one band in our gel on the SDS page of a specific size that we estimated, we likely have our purified protein. Now that does it for more of the wet lab stuff. Just to cap things off, we added all our design parts to the registry. You can even see them now if you go online. This will include our coils and our binding proteins. Now, because neon green and cherry were already available in the registry, we were able to actually create some nice composite parts for our fluorescent binding constructs, and all of these are RFC10 compatible.
The device itself is comprised of a microneedle array collection component, which is placed onto the patient's forearm to capture interstitial fluid, or ISF, by capillary action. Fluid is then transferred to a fluorimeter-type device with fluorescent binding-rich gold-plated chambers to capture each metabolite of interest and ensure binding with the coiled coil cysteine tails. A light then replaces the microneedle patch to measure fluorescent intensity with a detector housed in the device body, which communicates the metabolite concentration to the user through an accompanying app. Note that the system contains multiple chambers, one for each analyte detected, with a revolver-like design enabling convenience and cost-effectiveness. The user interface for an accompanying mobile application is depicted here. This app would be compatible with the biosensor to interpret levels of hormones and minerals alongside other critical and trackable factors for CKD patients. These include mood, activity level, blood pressure, meal details, bowel movements, weight, pronounced symptoms, and any changes in medication. This would consolidate the patient's data to be interpreted by their healthcare provider for a more holistic approach to care. Patient-centered care is an important new direction in modern treatment, and cannot and should not separate mental health from physical health, in addition to these other related factors. For example, weight tracking can help users with diabetes, which is a very common comorbidity to CKD, measure fluid retention, which can be related to blood pressure and further complications. Modeling was conducted to analyze the flow of interstitial fluid through 25-gauge microneedles, determining that a 15 to 20-minute collection period would be required to collect 287 nanoliters of fluid, as depicted in this graph. This is reasonable given the minimally invasive nature of our collection method. This flow rate calculation accounts for ISF flow through the dermis and then uptake and flow in the microneedle itself, as can be seen in the microneedle schematic on the left. Flow is characterized by determining important non-dimensional numbers, such as the Reynolds and Bond numbers, using fluid mechanics equations. The Hagen-Fosset equation was used to estimate the rate of flow of ISF through capillary action, confirming that it would be a sufficient driving force for the device, such that additional vacuum suction methods would not be required. The Lucas-Washburn equation is shown as an example here, applying Fosset's law to estimate the pressure drop in ISF across the needle.
Throughout the summer, we heavily engaged in synthetic biology education, creating seminars and essay contests for high school students to promote more interest in synthetic biology. We also have several other SynBio education related outreach activities in the works right now that you can look forward to at the final cGEM presentation. We also formed a very meaningful collaboration with the University of Rochester and Lambert High School, where we formed a biosensor working group and shared critical project advice, troubleshooting, and overall support for each other's teams. We also give a big thank you to all the cGEM teams in Canada who we frequently collaborated with over email, Instagram, or Slack. You also may be wondering where our human practices work is, and rest assured that is in progress right now. We are currently sorting out confidentiality agreements with a lot of our advisors and people we spoke to, along with finishing up some final projects. A big thank you to the phenomenal Queen's cGEM team who have worked so hard over the summer despite the challenging circumstances we're all in. And thank you to all our advisors and our funding partners. Thank you.